For the Calvin cycle, CO2 enters through the stroma. The rubisco catalyzes a reaction between the carbon dioxide and RUBP and forms the 3-phosphoglycerate. The ATP and NADPH use their stored energy to transform the phosphoglycerate into the glyceraldehyde 3-phosphate. The ADP and NADP plus result from this reduction process. One of the glyceraldehyde 3-phosphate molecules leaves the Calvin cycle and will form glucose. The rest of it moves on. The remaining glyceraldehyde 3-phosphate and ATP are used to regenerate the RUBP. For the light reactions, I kind of based mine off of figure 14.32 and just kind of read it a little bit. The light comes in to the photosystem 2 and strikes the chlorophyll molecules. The oxygen evolving complex basically splits the water and then forms oxygen and hydrogen. The chlorophyll molecules send their energy to reaction center chlorophyll A. And then after that, the chlorophyll A donates its electrons to the electron transport chain. The plastocyanin and I'm not sure how to say that, form part of the electron transport chain, and then the electrons continue to move from photosystem 2 to the chlorophyll A in photosystem 1 via the electron transport chain. The cytochrome B6F complex is one of the links in the chain. And then after that, the reduction center chlorophyll A donates its electrons to the ferredoxin. And then the ferredoxin donates its electrons to, I forgot to write it down here, the NADPH and the NADP plus. And then the NADP plus uses these electrons to make the NADPH. The ferredoxin donates its electrons to the ferredoxin NADP reductase, which is down here. And then the ATP synthase brings the protons back across the membrane and then turns them into ATP. And then, just a side note, the ATP and NADPH are both made in the stroma, and that pretty much wraps up the light reactions.